Chapter 6 deals with a review of the various immune tissues of the body. Fundamentally, we need to decide or distinguish immune tissues into primary lymphatic organs versus secondary lymphatic organs. The primary lymphatic organs, which are sites of B or T cell maturation, respectively, are the bone marrow and the thymus. The secondary lymphatic organs are lymph nodes and all of the related lymphatic tissue associated with lung and gut, as well as the spleen. First, let's deal with actually the flow, drainage, and formation of lymph. The lymphatic system basically drains extracellular fluid that's found outside vascular tissue in the extracellular space of most organs. Lymphatic capillaries begin as blind tubes that are lined by an endothelium that has an incomplete basal lamina and lacks tight junctions. These lymphatic capillaries drain progressively to larger lymphatic vessels, which have one-way valves somewhat analogous to those of veins basically in the limbs. Larger lymphatics basically will also have some surrounding smooth muscle. Lymph is transported by lymphatics first to lymph nodes, then traffics through those lymph nodes from one node to the next, and ultimately back into the venous circulation through either the thoracic duct, which drains into the origin of the left brachiocephalic vein, or through the right lymphatic duct that drains into the origin of the right brachiocephalic vein. As we know from gross anatomy, the thoracic duct is draining much more lymph because it is draining all of the lymph from tissues below the diaphragm. Lymphatic vessels are not found in the central nervous system, cartilage, bone, bone marrow, thymus, placenta, cornea, teeth, or fingernails. First off, let's look at some of the cells that characterize various components of the immune system. First at the top of the list in this chart are part of the cells in the myeloid lineage, including monocytes. Monocytes are famous because they give rise to various phagocytes, including macrophages. And typically, a monocyte will have a horseshoe-shaped nucleus, and these are some of the largest cells, which we'll see in a peripheral blood smear. Macrophages, as we just said, are differentiated from blood-borne monocytes, and they typically are the cells that are phagocytes. They're also involved in the secretion of cytokines, and in addition, they serve as one form of an antigen-presenting cell. Dendritic cells are another example of an antigen capture, transport, and presenting cells that are going to be characteristic particularly of lymph nodes and the spleen. Neutrophils have a classic multi-lobe nucleus, and they're kind of classified on the lack of staining of their specific granules. They tend to be small and pink. These cells and their specific granules are mainly involved in the phagocytosis of bacteria. Eosinophils, characterized by the presence of large pink eosinophilic staining specific granules in their cytoplasm, are associated with antiparasitic reactions, basically involved in the killing of antibody-coated such parasites. These eosinophils, instead of being multi-lobed, tend to be bilobed. The least frequently seen of the blood-borne white blood cells are basophils. These are like the connective tissue mast cells in that they are associated with granular release in conjunction with active substances that are produced during allergic reactions. Mast cells are equivalent. There's some debate about what is the common lineage associated with these two cell types. Mast cells are typically found in most connective tissues, mucosa, and adjacent epithelia, and they again are associated with the release of granules mainly containing histamine that are associated with allergic reactions. Then at the bottom of the list are the classic immune cells, including lymphocytes, which have a dark round nucleus that largely fills up the cell, so there's a narrow rim of cytoplasm surrounding it. And certainly these come in two flavors. B cells are involved in antibody production. Cytotoxic T cells are associated with killing altered or infected cells that are to them. NK, or natural killer lymphocytes, basically are involved in the killing of tumor or virus cells, and they also target antibody-coated cells that are presented to them. Plasma cells are modified lymphocytes that are associated with antibody production. And the next slide illustrates in a single smear, although this is highly unlikely, fundamentally the 5 different common white blood cells that are seen in bone marrow and peripheral blood. The largest of them, certainly at the top, is the monocyte that looks simply like a large lymphocyte, but it tends to have a slightly indented nucleus and a larger amount of cytoplasm. And as we said earlier, these are the largest white blood cells that are characteristically seen. Number 2 is a classic view of a lymphocyte. You can't distinguish whether it's a T or a B cell. Note that the nucleus is always kind of circular or elongate, surrounded by a thin rim of cytoplasm. The remaining white cells basically have a lobulated nucleus, starting with a neutrophil that has a multilobulated nucleus. There are 2 examples here, one unlabeled at the top right of the slide, one labeled at number 3. Note that their cytoplasm is relatively poor staining because their specific granules do not react well in histologic stains. In contrast, it's very easy to distinguish a basophil at the lower left from an eosinophil at the lower right, labeled at 4 and 5. Note the dramatic staining of the basophilic granules in a basophil that virtually obscure parts of the nucleus. And in contrast, you can see the bilobed nucleus that's characteristic of an eosinophil with its characteristic eosinophilic specific granule staining. Finally, here's a histologic section of plasma cells, and they're unique because their nucleus, although typically rounded or slightly indented, typically occupies an eccentric position in the cytoplasm. And classically, plasma cells have what we refer to as a clock face array of peripheral heterochromatin, which makes them distinct. And they also have prominent amounts of basophilic staining because of the presence of rough endoplasmic reticulum. An electron micrograph of a plasma cell is shown next, and again, you can see the clock face peripheral array of heterochromatin at the nuclear margins and the prominent amount of cisternae of rough endoplasmic reticulum that characterize the cytoplasm. Next, in addition to macrophages, what are the other antigen-presenting cells? Well, they include a population generally of follicular dendritic cells that are going to be found in the spleen and the lymph node, and a specialized population of dendritic cells known as Langerhans cells that migrate into the basal layers of the epidermis. All 3 of these cell types are capable of picking up and presenting antigen mainly to T cells that have the appropriate T cells receptors that can recognize the antigen. T lymphocytes recognize antigen that's presented only with MHC1 or MHC2 complexes. These cells can proliferate, form cytotoxic T cells, or form cells that promote or suppress the immune response of antibody-producing B cells that recognize the same antigens. B lymphocytes give rise to long-term memory cells that will leave lymphatic tissue, such as the spleen and a lymph node, and circulate throughout the body. They certainly also give rise to antibody-secreting plasma cells that we illustrated earlier that usually secrete their antibody locally and do not circulate basically in the peripheral blood. They're mainly found certainly in connective tissue and in various immune-presenting and in immune organs.